So we'll move to our second speaker, Dr. Yu Shi Will Wang. So I'll introduce you. Meanwhile, Dr. Wang is an assistant professor at the Center of Genetic Disorders and Aging with the Stanford Burnham Davis Medical Discovery Institute. Dr. Wang earned his PhD in stem cell biology at the University of Ottawa before completing a postdoctoral fellowship at Stanford University School of Medicine with Dr. Helen Blau. During his postdoctoral fellowship, he collaborated with Gary Nolan's lab, focusing on skeletal muscle regeneration, aging using advanced immunofluorescence techniques. Today, Dr. Wang will share his discoveries of mechanism of peripheral motor nerve regeneration in his talk titled Spatial, Regulators of Neuromuscular Regeneration and Aging.
Right. Thank you. This? I use this? Okay. All right. Thank you so much for inviting me here, Alex, and the organization of this wonderful symposium. I think it's been a great day of learning about spatial biology. You heard from Michael earlier about skeletal muscle. So you'll see that. What is the... Oh, found it. There we go. You know that skeletal muscles and the nerve connections to the skeletal muscles themselves are very important for everything that we do, you know, from breathing to moving to being able to stand to your eyesight. You know, you have little muscles that control how the shape of the eye is. And when these things go awry, either in genetic disorders, as the ones that Michael mentioned, including Duchenne muscular dystrophy, spinal muscular atrophy, you know, it's devastating to see a young child rapidly lose their muscle function and then deteriorate into, you know, being requiring a wheelchair and eventually premature death. And as we get older, muscle function is lost inevitably as people become frail. As you can see here, using this MRI capture between a young versus an older gentleman, a 40% decrease in muscle mass across a few decades of life. And after about the age of 50, you know, 5% muscle loss per decade is kind of a metric that occurs to both males and females. And so when I was trained as a stem cell biologist and looking at tissue regeneration, muscle regeneration in particular. And so when I think about, you know, genetic disorders and chronic progressive degenerative diseases, I think about it in the context of regeneration. And so if you think about how a young skeletal muscle regenerates, it's not just a stem cell activity to replace the functional tissue. You also have concurrent processes in inflammation, fibrosis, and stromal cells, and eventually innervation that play a major role in restoring a functional tissue. And as you think about degenerative disorders, various processes become dysregulated. It's not just the stem cells that become dysfunctional. You have inflammatory processes that become dysregulated, tissue fibrosis, altered tissue stiffnesses can also change how cells behave. And so I like to think of, you know, the different cells in the tissue as almost like an ecosystem. You have different roles. The cells have different niches. There's relationships and dependencies. And for us to map those relationships, we really have to think about the tissue microenvironment, how these different layers of processes and extrinsic signaling get integrated into the cell. And not only at the transcriptome level, epigenetic level, but even at the level of, you know, stromal and vasculature, extracellular matrix, but also histone modifications, protein regulation at the post-translational modification level. And so when we look at the protein, our collaboration with Gary when I was at Stanford really led us down to kind of doing this by using Codex, which is a highly multiplex immunofluorescence platform, where a panel of primary antibodies are conjugated with oligonucleotide barcodes. And so when we validate this set of antibodies and the barcodes themselves, we can stain a tissue section, and these are thin sections, with the entire antibody panel up to 55. And I know Akoya is now able to do more than that. And then what we do is we place this onto a microscope with a microfluidic system that's able to inject complementary fluorescent probes 3 at a time. We reveal them, and the imaging is rapid, and we could do this over the course of a few days. And then, once those 3 probes are imaged, we do 3D stacking, stitching, and registration across cycles. We can remove those probes, and then the tissue becomes autofluorescent again, and then we anneal new probes, and then we image those 3 barcodes. And then so you subsequently reveal all of the barcodes and all of the cell types that you're interested in. So by having a compendium of every cell type in the tissue, you really get a lot more information into the how the cells relate to each other. And by having the ECM information on top of that, you can see how the stromal architecture impacts the cell function. And so this is what a muscle looks like. This is a mouse tibialis anterior muscle sectioned longitudinally along the muscle fibers. So the muscle fibers are going along in this direction. As Michael told you, they're some of the largest cells in your body. They can be centimeters long and have 1,000 nuclei in them. So they're very interesting cell types that contract and experience a lot of physical stress as well. And so you can see the ECM structures around them in their basal lamina and lamina. The muscle, of course, skeletal muscle is connected to bone. And so you have the tendon that connects the muscle fibers to the bone. And this is a very complex ECM collagen fibrils that make up the tendon. And so you can imagine how much force needs to be translated in order for you to lift something, right? And you can imagine a bodybuilder lifting like 200 pounds, that is a lot of force being translated. Of course, skeletal muscle is innervated. And so we can see the junction points where the nerves come into the muscle. The nerves themselves actually live in the spinal cord, in the lower spine, lumbar spine area, and they send out the long projections that go all the way down to your muscles in your lower limb. And so you can see that those axons can be almost a meter long. And imagine that in terms of, when you get a peripheral nerve damage and having to regenerate that entire length of axons. Of course, there's tissue-resident immune cells. The muscle is very well vascularized, and there are great interactions between the cardiovascular system with muscle itself as well. And of course, when we do a controlled injury, this is with a chemical induced injury, you can see how much architecture is disrupted. But actually, the muscle itself is able to regenerate very well. And so in these types of acute injuries, this is a healthy muscle. Now, cross-section, so individual muscle fibers are like this. You can see that the entire architecture can be completely disrupted. But over the course of about 2 to 3 weeks, the muscle fibers come back, regenerated. And actually, the stromal architecture is almost all completely restored as well. But in chronic injuries and disorders like progressive myopathies and in aging, you get collagen deposition, fibrosis, and persistent inflammation. So this is really a question of instructions. How do these cells talk to each other? How do they form and use a blueprint, a genetically encoded blueprint to rebuild a tissue, right? And so during my time as a postdoc, we did a very well-characterized study with, doing single-cell RNA sequencing, single nuclear RNA sequencing to map out the different cell types, identify antibodies needed for this type of approach to map all the different subsets. We developed algorithms to do image registration across imaging cycles that are better than current available platforms, and then also segmentation, improving segmentation at the nuclei level, but also at the muscle fiber and cell type level. And then improving kind of machine learning approaches to cluster these cell types using multiplex IF data. And so we were able to really rapidly generate from across multiple tissues an atlas of tissue regeneration. And so you can see that here. We imaged about 30, 40 tissues and looking at a dynamic time course of all these different cell types over the process of tissue regeneration, identifying cellular neighborhoods. I'm not going to go into that too deeply, but we've also done this, looking at translating this from single cell RNA sequencing in articular cartilage, looking at chondrocyte heterogeneity, and then translating that into looking at how these different populations identified by single cell RNA sequencing actually map within the tissue, and actually seeing kind of layer structures that are revealed by transcriptomic data. But today I want to focus a little bit on molecular drivers in muscle aging, and how can we actually target some of these pathways that are normally regulating tissue maintenance and restore muscle function in aging. And actually that resulted in us understanding gaining a better understanding of how nerves actually regenerate to the muscle. And so when I started as a postdoc, we ended up identifying this immunomodulatory molecule called prostaglandin E2, which is needed for stem cells to activate. And this is actually been described for hematopoietic stem cells, neurostem cells, and also muscle stem cells and intestinal stem cells. So it's a pretty common pathway. And actually prostaglandin E2 is that fever molecule that you get when you have inflammation, you get a cold, the flu, you get the fever, you take NSAIDs to suppress prostaglandin E2 signal. It's also known as the pain molecule because that's a part of the nociception pathway. But for regeneration, this is a signal that activates stem cells. So your body senses damage and then triggers the stem cells to activate. And we initially thought, okay, great, no pain, no gain, of course, right? And so what happens in aging? We initially thought this is clearly something that's going to be chronically activated because this is something that is common to aging inflammation. But when we actually did the mass spectrometry analysis in young and aged muscle, we ended up finding actually prostaglandins in general were decreased at the tissue level. And this was actually due to an increased expression of the enzyme that degrades prostaglandin E2, which is called 15-PGDH. A dehydrogenase that uses up NAD to metabolize prostaglandin E2. We used Codex to overcome some of the issues with imaging skeletal muscle and aging, a lot of autofluorescence, tissue autofluorescence that accumulate, but because when we do Codex, we can capture the tissue fluorescence before we capture the antibody signal, we're able to subtract that background out, and we were able to confidently show that 15-PGDH gets upregulated in these aged muscles. What was kind of striking is that when we use shRNA delivered by AAV into the muscle or using a small molecule inhibitor against this enzyme, over the course of a month in 24-month-old mice, so that's the equivalent of like a 70-year-old human, over the course of a month, the muscle mass and muscle strength was restored to almost levels of young within a month. That's pretty exciting. And so the other part of this was when we overexpressed this enzyme in young mice, the muscle shrunk away. And so we termed this enzyme, and there are other classes of this, potentially a gyrozyme, something that's upregulated in aging, disrupting something useful that's needed for proper tissue function. And so we really wondered, what is driving this in aging? What could be possibly turning on a molecule that just kind of allows the muscles to waste away? And we thought about this from the connection of nerve and muscle, and because the nerve is very important in maintaining muscle function, does this have something to do with the upregulation of 15-PGDH? And we know from historic data from Josh Shane's group at Harvard and others that the neuromuscular junction, the connection between the motor nerve and the muscle, becomes partially affected through neuropathic processes with age. And when we look at neuromuscular junctions in young mice, they have this classic pretzel shape with synaptic connections that perfectly overlap the acetylcholine receptors on the muscle. But in aging, these connections become completely dysregulated morphologically and become disconnected as well. And one of the things that really drove us to thinking about denervation was when we looked at the tissue and where 15-PGDH in yellow here is expressed, Now, when we look at across the muscle fiber in different muscle fiber types, whether it's fast twitch or slow twitch muscles, the 15-PGDH was preferentially being upregulated in the fast twitch muscles. And what we know from historic data is that the fast twitch muscle fibers are more affected by denervation and aging. So this is a very partial process that affects fast motor neurons that then only affect the fast muscle fibers. And we can see this gradient. So, the muscle fibers that are closer to the bone are slow twitch, and the muscle fibers that are more superficial, closer to the skin, are faster twitch. And you can see that the gerozyme upregulation is very much localized to the superficial areas. And so we took a step back. We thought, okay, if this was the case in one muscle group, can we see a general faster twitch muscle group versus a slower twitch muscle group, which is the EDL is the faster and the soleus is the slower. And we look at just by Western and proteomic analysis that actually the upregulation of our gerozyme very nicely correlates with fiber type. And so we said, okay, this is what's happening naturally in aging. Can we model this actually with a nerve injury? And so we performed a sciatic nerve transection, and by both single nuclei RNA sequencing, and also by protein level at the using codex, we can see that the denervated muscle fiber actually upregulates this enzyme. And so this is great. This is mouse aging, mouse nerve damage. Do we see it in humans? And so we contacted a pathologist, our colleague at Stanford, and said, okay, we need samples of human neuropathy. Can we peripheral neuropathy? Can we get some muscle biopsies? And there are a large range of muscle disorders that affect the peripheral nerve that ends up causing muscle wasting. And when we stained these tissues, and so pathologists look for these muscle fibers that have a very interesting histological phenotype. The muscle fibers themselves are heterogeneous in terms of their mitochondrial distribution. The inside of the muscle fiber loses their mitochondria, just disappears through a mitophagy mechanism. And so when we stained these tissues, we found exactly where the mitochondria were missing, we found our enzyme. And it was kind of striking. And when we did serial sections to look for autophagy mechanisms and where the mitochondria are localized with pyruvate dehydrogenase here and LC3A as a marker of autophagy, you can see the nice correlation of our enzyme with autophagy mechanisms, but also mutual exclusion of mitochondria. Okay, so that's kind of, that shows you that this is also active in human myopathies. So what does this enzyme do functionally? Well, we performed a peripheral nerve crush injury, so kind of more similar to something of a trauma that is reversible, not a severing of the nerve, but something that can regenerate. And so the first phase of this, the muscles lose their strength because they're no longer connected to the nerve. They waste away at first, but then they regenerate over time. And so for the first week, we can see that the muscles are wasting away. But during this time, when we treat with this drug, you can see the regenerative phase is rapidly accelerated. And this is not just at the mass level. We're not just getting more muscle. We're actually getting better connection of the nerve and muscle. And how we know this is we've actually taken sections of the peripheral nerve post the site of crush, and we stain for acetylcholine receptors to mark the motor axons versus the others. So we train a neural network to quantify this because nobody wants to count 4,000 axons. And it's very compelling because we seem to have a selective restoration of motor axons, whereas the other axons are not coming back as quickly, which is fascinating to think about. And of course, when we look at the level of neuromuscular junctions, we can see that at 2 weeks after treatment, the nerve connection is actually established. And so it's not just axonal regrowth, but also the synaptogenesis. And so this is our current model for this process. And we do think, and so we tested this in aging to see if the muscle function restoration that we saw in aged mice is related to neuromuscular regeneration. Indeed, there was a decrease in most of the pathological phenotypes in neuromuscular junctions of the aged mice after 1 month of treatment, and that is concurrent with a 15% increase in the strength of those mice. 15%, as I said in the beginning, 5% per decade. 15% in a month is able to recapitulate quite a bit of the loss that you get with aging. So fascinating, right? So you have this enzyme that's upregulated in the muscle, right? And you get this phenotype in the spinal cord and the nerve regress. So we thought about this for a moment. You know, chances are you're modulating prostaglandin levels in the muscle. And how is this connected? So we actually just did a single injection of prostaglandins in the lower leg, right here, right? We wait an hour, and we take out the spinal cord, we sack the mice, take out the spinal cord, and we look at the motor neurons in the spinal cord now. And you see that so prostaglandin signal through prostaglandin receptors that then signal through cyclic AMP. And now when we look at the cyclic AMP response element binding protein, so CREB, phosphorylation of CREB, which is the active form in the spinal motor neurons, within an hour from the muscle to the nerve, you can get this rapid activation of CREB. And in aged mice, this is particularly striking because after that 1 month of treatment, we saw fewer cleaved caspase 3 positive motor neurons in this spinal cord. So suggesting that we're actually blunting the neuropathic process related to aging, at least in the peripheral nerve. And so this is our current model of what we think is happening. Prostaglandins can act not only at the stem cell level, but also at this crosstalk between muscle and nerve. And so with aging, with this pathological process of denervation initiates a positive feed-forward loop. You have the upregulation of 15-PGDH in the muscle, which then also downregulates this pro-regenerative molecule prostaglandin E2 with the nerve itself. And so my lab is now kind of thinking about this, not just at the tissue, not just at the muscle level. We're really thinking about how immune cells, the nerve, and the muscles interact in this whole degenerative process. And so we're really taking this to a multi-tissue interaction level, so we can look at kind of the immune cells inside the bone marrow, inside of splenic tissues. And then also, actually, they're actually pretty close to each other. If you look at this, this is the bone itself, this is the muscle, and these are the tendons next to it. And so the tissues are not actually that far from each other, even though we're thinking about systemic regulation. And we're able to zoom in with highly multiplexed immunofluorescence. You can see all the different cell types in the bone marrow. And apart from that, we're looking at epigenetic regulation, cell type phenotyping on top of that layer as well. And so this is my lab down in San Diego, except for Bernard Previs, where we have a lot of collaborators. We're a very collaborative group with lots of people around the area and across the world. So with that, take any questions.
Thank you for a very interesting talk and being on time. Do we have any question in the crowd?
That's really awesome. You sort of touched on this at the very end. So is this prostaglandin dehydrogenase inhibitor, is it working on the myofiber or on the muscle stem cell, or is it working on the motor neuron, or is it working on the myofiber and then insulin? Like, so is it working on everything? Like, how's that going?
Yeah, so it's good question. So it is kind of pleiotropic. So the inhibitor itself, because this enzyme is not just expressed in the muscle, it's also other places. So there are other effects in other tissues as well. And people have reported this on the hematopoietic system as well. But in the muscle system, the enzyme's expressed in the muscle, but prostaglandins are kind of, signaling molecules that influence other cell types, right? And so you inhibit the enzyme in the muscle, and that's increasing prostaglandin signaling to other cell types that interact with the muscle. So it's like, you fix the muscle fiber, And then the surrounding cells that connect to the muscle fibers, that support the muscle fibers, realize, hey, we need to reestablish our connection here.
So is the receptor, prostaglandin receptor, is it on the axon of the motor neurons?
We're trying to figure this. It is expressed, yes. So actually, in the paper, we showed that it is expressed in there, especially the cholinergic neurons. Yes. Huh. Yes. So at least we're getting into looking at because the pathway to the FDA is not that easy through aging. And so, of course, genetic motor neuron disorders are kind of what we're considering. And so, of course, ALS models, at least in mouse, we collaborated with Doug Malay's group in Cincinnati, who's been doing single nucleotide characterization of these mice. And there does seem to be upregulation of these the enzyme, at least in the SOD1 model. And we've looked at SMA models as well, and it's the same case. Yeah, and actually, some of those tissues that I showed in the hemomyopathy side, the motor neuron disease, it actually was per patient.
I'm just wondering, do you know what's really approximate information?
Well, so this this is this. I mean, it's a very small molecule, right? It's a lipid. It can go through the systemic circulation. So if you consider, you know, you have a infection somewhere, it can cause fever and throughout your entire body. So it gets signaled to the brain pretty rapidly. So I don't know. I think the range is systemic, but I don't know. It's very difficult to measure because actually it's degraded very rapidly in the body. So it's like, you know, if you do pharmacodynamics, it's like minutes. The half-life is minutes, 3.5 minutes. Yeah.
So I have actually two questions. So first of all, because we are in a spatial biology symposium, I was wondering which of omics that you use, the spatial biology methodology that you use were more informative in your studies, and would you recommend that people who want to look at single cell or more high resolution changes?
Yeah.
The second question refers to the immune response, because you were talking about inflammation, and I was wondering, what is the insight? Do you think that this enzyme is coming mostly from where? From immune cells? Is it innate immune cells? Are those microglia, or are those peripheral monocyte macrophages, or is there like other type of cells, adaptive or innate immune cells that are involved here?
Yeah. Okay. Those are great questions. So the first, I'll go to the first one. So the omic, in terms of omic technologies, we're doing spatial proteomics using antibodies, so it's targeted. We know the targets that we want to identify using by doing single nuclear RNA sequencing and by lots of preemptive studies. And so, you know, it depends on the biological question. I think really my advice to the audience is really depends on your biological questions, whether you want to go and study one particular molecules, in our case, you know, looking at how this is distributed, how this particular molecule affects this disease in tissue, versus, you know, unbiased approach like spatial transcriptomics. But at the end of the day, you know, you're the experts in the tissue, so you need to infer upon the technology what you need to do, right? What you want to look at. And oftentimes, I think what our advantage is being able to analyze our own data, drive those target kind of like discovery process, and then kind of go through that ourselves. And so it really requires kind of like this, you know, interplay between wet lab and dry lab and your computational abilities. Yeah. And for the second question, you know, the involvement of the immune system is very complex. For something like aging is just it's very difficult for me to answer that, because even between tissues that we look at, for aging. You get different phenotypes in terms of immune responses, whether innate or adaptive. And we do see an adaptive immune kind of B cell dysregulation that's affecting this. And we think that this is regulating neuromuscular connectivity. However, the cause and effect chicken and the egg kind of scenario here, we're trying to prove. So we're still working on that. But this enzyme particularly is expressed mostly by myeloid cells. And so it is expressed at least transcriptomically when we look at atlases by microglia, by macrophages and monocytes.